We're really delighted to have with us today the Nobel laureate, Dr. Helen. Thank you so much. Dr. Carrico is a biochemist and a vaccine pioneer. You all know this. She's going to share with us today her remarkable journey in science. She won the 2023 Nobel Prize in Physiology or Medicine for contributions to the development of the mRNA used to create the FDA-approved COVID-19 RNA vaccines by BioNTech, Pfizer, and Moderna. She shares this Nobel with Dr. Drew Weissman. Her 4 decades of research focused on RNA-mediated mechanisms with the ultimate goal of developing an in vitro transcribed protein therapy. Her work has widened the therapeutic applications and potentials of RNA. Dr. Carrico is a professor at University of Szeged, which is where she also received her PhD in Hungary. She's adjunct professor at the Perelman School of Medicine at the University of Pennsylvania. She's former senior vice president of BioNTech, co-founder and CEO of RNARX, served as CEO, a company dedicated to developing nucleoside-modified mRNA for therapy, and she's the co-inventor of numerous mRNA-related patents. And having spent the morning with her, I'm here to tell you she is a fascinating individual. She has an amazing story. If you haven't read her book, Breaking Through My Life in Science, it's brilliant. We're elated to have her here today at the Wistar Institute to celebrate her vast accomplishments and learn about her perseverance and resilience, and to let her tell you the story of her science and her life and life's work. So Catherine, we're ready to get started. And without further ado, I give you Dr. Helen Carrico.
Thank you. Thank you very much for this nice introduction. Just in case, this is the Nobel week, so I get mine. So if maybe tomorrow chemistry, you get yours, you can look at how it looks. At least the front is the same. So I will put on the desk there, and so don't take home. Just yes. So so thank you very much. This is a great honor to receive the award, and it is belonged to the outstanding women scientists who received this award prior years. Is really humbling, and I met them recently because recently I became famous and meeting famous people. But I have to tell you that I am still today, I am just one of you. I usually sit there and listening here, great lectures. I cannot guarantee today that it will be that scientific because I thought that I am talking about more like my journey. You can look at so many YouTube video about the science itself. So I read about Helen King. I did not know what she accomplished, and not just the Wikipedia. That Bonnie sent me an article that how she was ignored at the beginning and not acknowledging her work to create the rat. I have to say that I work in neurosurgery 17 years. We work with Wistar rat there, but I didn't know the origin and never thought about that somebody had to create. But you have to know that even a Eppendorf tube, somebody has to come up with the idea and the license and so on. So my journey started far away from here in Hungary, and I grew up in a very simple household. We didn't have television or running water and many other things, but I didn't even know that it could be because none of our neighbor had one, so I didn't miss anything. And I had a sister who is, she's 3 years older, and if you have older siblings and they are doing good in school, you go to school and they expect you to be good one. So that's what I started to work and study very well. And I already, as an elementary school, I participated in science competition, chemistry, geography, but biology I was more successful. And when I was 14 years old, I was third best in Hungary in biology. And so that we went to those who participated in competition, and we went to summer camp. And here you can see these girls. I have a cursor, can you see? Oh, no, that's not there. Okay, so that's the tallest one there is me. And some other girls became biologists, physicians, chemists, later life I met. But here everybody's 13 years old, these girls. And from elementary school, I went to high school, and I mentioned this because I had excellent teachers, and because we couldn't speak English. You see, my English I started to learn in university. So anyway, we could write only Hungarian, and so we selected Hungarian famous scientists and sent them letters. And most importantly, we sent János Seje, who is a Canadian scientist, is Hungarian origin, and he coined the word in the 1930s, stress. Prior to that, it was only in the physics. And he wrote a book about how to avoid stress. And it was translated in Hungarian. And I wouldn't stand here today if I would not read that book when I was 16. So I learned how to handle stress. And that's why I could survive in this very stressful environment. Anyway, so he worked also with rats, actually, and he had low budget, like always in my life. And he chased the rat in his office with a broom, and he demonstrated that the brain can be damaged. And he demonstrated that the stress can kill us, but not the stress itself, but how you perceive it. And you can learn to perceive differently. Even the worst things, you can turn into a positive stress. And positive stress is, the excitement, the anticipation, which we felt this morning, that we will gather here today. So the main thing is what he said is that you always, what the Stoic in ancient Greek, Rome said, you have to focus on what you can change and not what, other people should do, because that frustrates you. Your grant is not funded. Those people didn't make the right decision. Your superior is somehow, advancing somebody else. Somebody else get the pay raise and so on, and you get frustrated because people are not doing, your wife, your husband, your children, your neighbor, nobody's doing what you want them to do. And that's the stress. So you have to figure out what you have to do so that everybody will do what you wish them to do. So that takes time and practice. Anyway, so in high school, when I was in high school, I had a program similar. I learned here that Vistar has educational program. So the university advertised for those children whose parents were not highly educated. My parents had only elementary school education. So actually on the very right one here, that's me here. It was in the newspaper I cut out. And so we could go to the university because I have never seen a university. I never seen a professor. So it helped me that when I went to the entry exam to the university, not this first time I see a professor, which looked like, just anybody else, but that's how it is. And so my dream was to go to this University of Szeged. And the reason is because on that year, they just opened this huge biological research center. So it was 50 years ago. And actually, a professor at University of Szeged, Albert Szent-Györgyi, who got the Nobel Prize in 1937, while he was teaching at the university, he opened it. I couldn't attend because only the selected people, but 50 years anniversary, I was the one who celebrated that. Anyway, so that was my dream, to work in this institution. So as a student, undergraduate student, I was eager, and I went there to find a job, maybe, just like a student, to do something with my hands. And genetic department, all of this excitement, but the only place was opening is the lipid lab. Even today is lipid kind of boring, kind of, but But, you know, the lab had explained to me that actually they just discovered that this membrane is not really how I learned in high school, you know, layer of protein and lipid and protein, but that fluid mosaic model was just introduced. There had excitement. And I immediately learned that whatever I learned in high school actually is out. You know, these things are not like that. And so I was so eager to spend the first summer in this nice institute and investigating lipids, but Tibor said that, hey, Kati, you can be the rest of your life in a bench. Go and go a field trip, you know. At that time, you know, I wanted to at the bench. I didn't know that at age of 58 here, you know, in the Stemler building, I am still doing the experiment at the bench. I didn't know. So anyway, I was a little bit disappointed now that I had to go to this fishery institute and collecting water fleas. You know, that you can have the big dream, you will be the big scientist, you discover vitamins or something, and finally, you know, you collect little insects in the water, doing a little experiment, collecting fat from fish. But no matter what is, you know, you just, you know, dig into and try to learn as much as possible. And so I spent there a summer, and when I came back to the laboratory, fellow colleagues, Ernő and Eva, approached us that they need phospholipids, phosphatidylserine, phosphatidyletanolamine, but we were behind the Iron Curtain, and we could not order. This was a couple of Deutschmark, that time no euro yet, but we couldn't order. So we looked it up that how we could have this, and this was from, we could isolate co-brain, and there was this paper described in 1942. And so Ernő went to the slaughterhouse, that's the PI at that time were doing, you know, with the bike, and came back with a co-brain. And I didn't mention my father was butcher, so I was not really bullish about seeing a brain and started to work, isolate lipids from it. But this is again a lesson that when you want to do something, you can find a way. If not, you can find excuses. Okay, so that we find a way. And that was the first, you know, as an undergraduate, we're delivering with liposome DNA to the cells, and then the encoded protein was expressed. One day, a laboratory entered Jenő Thomas, and he told me that if I want, I can work in his RNA lab. Yeah? So you can see, I am not the visionary person figuring out that, oh, this is good in the end of 70s and the 80s, working with RNA and liposome, and then one day it will lead to something. It was just by chance. Okay, RNA. He told me that this molecule was just discovered, this 2', 5' oligoadenylate. This is responsible for interferon-induced antiviral effect, and we will make it. We need an antiviral compound. A fellow graduate student was János Ludwig, and even today I work with János, and that's what I suggest to you also. Be nice to your fellow students, you know, because you need their advice. Everybody was organic chemist in the lab. I was the biologist, had to set up a viral screening lab, and I also had to synthesize. So this picture of me when I was in 20-something there, it was really a Büchi, and I synthesized chemically these molecules, so it was not a setup there. You know, and with János, we published this because in the meantime, our supervisor went to Utah and spent there a couple of years. So we were just students in the lab. Anyway, eventually, this antiviral compound, you know, we couldn't make it deliver feasible way. And this was, I was at age 30 when I, you know, first time I was terminated in my position. And again, I wouldn't be here today if several times I would not be terminated. And so important things is, instead of feeling sorry for yourself when you get this very stressful knowledge that you have to leave, you know, focus all of your energy, what next? So I searched position in Hungary. I couldn't get, never even answered my letter. And then I tried to find a position in Europe. They responded, bring the money, but we were behind the Iron Curtain, so I couldn't. So, and finally, I had to come to Philadelphia, to Temple University. Okay, I never wanted, okay, many colleagues, you know, they wanted to come to America, but I was not one of those. I was very happy. But here, Professor Suhadolnik also worked with the 2' 5' molecules. We did different modified nucleosides. Actually, the cordycepin, which is 3' deoxyadenosine, is easy to make 2' 5' because you have just 2' hydroxyl there. This was actually in my thesis. I used these molecules and did such 2' 5' molecules. And yeah, so the budget was low. You can see no lab coat there, but you know, but we did a lot of studies, but eventually we also could not make this 2' 5' molecule to be a good antiviral molecule. And we needed one. We are in 1985, and the major problem was HIV. And we collaborated with the University Hahnemann that was not exist anymore, part of Drexel. So Hahnemann University, we collaborated and we tried to help the HIV patient. Interferon was discovered 1957, 1963 already. They knew that double-stranded RNA can activate and induce interferon. So scientists tried to use, you know, the double-stranded RNA to treat patients, but it was this double-stranded RNA was very toxic. If they gave more toxic, if less, it was not useful. So we made this mismatched double-stranded RNA, which still could activate interferon, but was less toxic. And we treated 10 HIV-infected patients. That was not helping them. Later turned out that they have a system, that many of the viruses, you know, have system against the treatment. So this was second time I lost my position. If you read the book about me, you know that it is quite a journey was. I ended up at Bethesda and commuted from Philadelphia. And then I came to University of Pennsylvania in 1989. Elliot Barnett, and a young cardiologist, he's as young as he was, just a week younger than me. So I convinced him that, hey, Elliot, we should, you know, use messenger RNA for therapy. And so because he was not knowledgeable on that, he said yes. And then he, you know, he was investigating urokinase plasminogen activator receptor. And this is a highly modified molecule, glycosylated, GPI link. So it is post-translationally highly modified. And what was important that we delivered the RNA, and then the cell knew what to do, how to decorate, how to do all of this processing, and made functional receptor. And that was when we realized that mRNA will be good for something. And so we were not the first one, of course, here doing all of these experiments. In the 90s, other laboratories already did therapeutic use of mRNA. For influenza, they did animal experiment. Influenza vaccine, they did. Even self-amplifying RNA was used, cancer vaccine. And I followed very closely the literature. You can see every year there was just one, so it was easy to do, not like in these days for you. But these people did not publish again. I mean, Floyd Bloom was, you know, editor of Science, and from Scripps, they published that how vasopressin mRNA could treat an animal. And for me, this was the major paper that decided that I will use mRNA, which will called therapeutic protein, and I will use it for treating. When I was in cardiology, you know, heart failure and intima thickening. And then later when I moved on, then whatever was the topic, I tried to use, but they never published. None of the co-author ever published on mRNA, but maybe that was the purpose of their paper, just to inspire me. And other people like the papers that on influenza vaccine. I asked scientists recently, and they said that they couldn't get funding and they stopped working. So Elliot, 1997, he left the laboratory. By that time, I was demoted from the faculty, which was, research assistant professor. You get this one in clinical department, PhD, just get the green card. Now I get the green card, and I was demoted, and Elliot left again. I was out of job, but David Langer, who I met in Elliot's lab, David was just a medical student, the medical student in the lab. If you have experience, they know everything. But I was still nice to David. And by that time, when I had no job, he was in resident in neurosurgery. And he could convince the chairman that neurosurgery needs a molecular biologist, and the chairman was convinced. And that corner, you can see that lab, 14 years I worked there by myself. And again, low budget, you can see no lab coat, not even gloves, but this an RNA laboratory maybe is use better things. And again, we want to invent something. And David said that how there is a problem, subarachnoid hemorrhage, vasoconstriction, and he can reach that vessel. And then I tried to come up with what kind of mRNA we could deliver. And it was like, we come up with nitric oxide synthase. And because the nitric oxide would focally just dilate the vessel, and it is millisecond half-life, but if we put the RNA, then it constantly producing the protein, and the protein would produce the nitric oxide. And we traveled everywhere, even Chicago, tried to find model system and so on, but David also left. But hopefully, or luckily, he visited time to time the neurosurgery department when they were close to terminate me again. And then he convinced them that I could stay up until 2013. But anyway, one day, I was coming back in Department of Medicine because Xerox machine was there and I knew the code, and I met Drew Weissman, who was standing there, and the new guy, and he told me that he just came from Fauci's lab. That really didn't ring a bell that time, and I was not on the field. So anyway, I told them that I can make RNA and, brag why he was Xeroxing there. And he said that he is interested in vaccines, therapeutic or prophylactic for HIV, and said, oh, I can do the RNA for you. And so that's how we started in 1997. And so he could make this human dendritic cells. He was telling me, I had no clue what it was. But dendritic cells, I thought that, something neuronal related, dendrite, but I have to learn again that whatever I learned in the university about immunology is out. And I learned all of the immunology from Drew, and hopefully he learned some RNA stuff from me. Anyway, he was very happy that everything worked well, the delivered RNA, we get a lot of antigen and so on, but we also get TNF-alpha. I was not happy. I was just wanted to treat a stroke patient with mRNA. And I mean, that's the last thing you want, more inflammatory molecule. And so we tried to figure out why the mRNA is inflammatory. And that's how we started, this experiment. By that time, I was working with mRNA almost 10 years. And so I had a collection of different isolates. And we tested out, interestingly, we found that not all RNA is equal. Some of the RNA, like the tRNA, was not inflammatory. And this, knowing that tRNA has so many modification, nucleoside modification, so came the idea that, okay, maybe nucleoside modification suppress immunogenicity of RNA. Okay, now just we have to make some RNA, messenger RNA, which contains modified nucleoside. It sounds easy, but as we know what we are doing, nobody has done how to do it. So there's, at that point, more than 100 modification in RNA molecule was known. I just showed those who are not chemists that to frighten them that many things. But, we always make our RNA from the 4 basic nucleotides, post-transcriptionally modification introduced by different enzymes. Yeah, but we couldn't call up Promega or Stratagene to send because those were not even known. Yeah, so that we have to make somehow differently. So then I call up Janos, Janos Ludwig, who were graduate student together, and ask him where I can bind modified nucleoside triphosphate. So he told me a couple of companies. One of them was very lucky because everybody later ordered from them anyway. So he told me that order. And I insisted just ordering naturally occurring because our body has to know how to handle. So we ordered these 10 and 5 of them incorporated to the RNA. So now we had messenger RNA with all of this modification. And when we tested out in dendritics, primary isolated dendritic cells, it turned out that some of them were not inflammatory. No TNF-alpha was induced. And when we looked at closer, we found that every case when the uridine was modified, the messenger RNA was not inflammatory. Who knew? Why the uridine? I don't know. But for us, it was important that the RNA has to be translatable. I want to use for therapy. Drew wanted to overexpress with viral antigen. So when we tested, we found that pseudouridine containing RNA is translates so much better. I don't have time today, the 2 years described that how we try to understand that why it is, the 2-thiouridine containing RNA not translated at all. So we selected the pseudouridine containing RNA and then as usually happening, we file a patent. And now that because I was already, had difficult time at Penn, so we established this company, RNARX, nobody heard about, in 2006 for, but our company could not get the patent. So, but we could get $1 million for this small business grant. So this is idea that if you have ideas, get the patent and apply for small business grant. At least it was good for 5 years, this $1 million, I could pay my salary writing check for Penn, and that's how I was employed. So here in this proposal, we said that we will treat anemic mice and demonstrate that we can increase with their hematocrit using messenger RNA coding for erythropoietin. So by that time, instead of the lipofectin, we used transit, which was working in vivo. And when we injected just 0.1 microgram pseudouridine containing RNA, even 4 days later, we could detect erythropoietin in the blood flow. And the half-life is about like 2 hours of this protein. So it continuous production was there. And if somebody works with nucleic acid, 0.1 microgram is very small amount. And it was physiologically active. So you can see in the middle panels that hematocrit was increased and weekly injection maintained high hematocrit in these animals. And finally, it was not inflammatory. So that was the last kind of work I have done. And that was, I was terminated at Penn and moved on to BioNTech, moved to Germany. Not this fancy building. No, we were in the campus and a handful of people. I couldn't speak German, so it was difficult because everything was written in German. And anyway, today, BioNTech is employing 10 years later, 6,000 people, but that time it was a small company, no website when I went there. And so we worked together, there. That was the plan with Ugur and Özlem, his wife, that they established the BioNTech. Before I was hired, actually, Ugur came to visit me. And I tried to make, collaboration, bring together my colleagues. And okay, in the Vistar, we were just in the front of the building. They didn't come inside, but I introduced Ugur, to Drew Weissman, and we went to the children's hospital Ugur gave lecture. There is Professor Morris there. And then later I invited back. So Drew visited many times and also get good funding from BioNTech, although at that time we had very scarce money, not much. And then also the Children's Hospital scientists, Jessica, Peter, and Adams were visiting and, helping, fostering collaboration. By that time, so 2014, we already improved so much on the RNA. So not this little protein, very short period of time. That was a significant amount of protein was made from the in vitro transcribed RNA because we modified the cap, the 3' and the 5' and modification and so on. And so the RNA was ready for prime time. So, the fastest growing section of approved medicine are proteins, but they are very expensive, the antibodies and other factors. So it would be good to using the cheap mRNA to replace those things and not only making cheaper, but accelerate the screening process and the protein would have the proper structure. You can make multivalent vaccine and others. You can put mRNA coding for 20 different protein and they will not aggregate because, the mRNA is always the fourth same nucleotide. So, and we can make intracellular RNA, delivering RNA and produce intracellularly the protein. So there's so many things we can use it. So I don't mention how many things I did. 9 years I was at Germany, but I mentioned this one, for example, we developed messenger RNA coding for bispecific antibody, and we demonstrated that how it can eliminate already established large tumors, and it is right now is in clinical trial. And so we reached the point now that you all remember as well as me, that what happened in 2020 when, we heard about that what's going on in China. And the quick action scientist in China, Yongzheng Zhang, published the data in January. Actually, he said it was January 6th. He put in the gene bank, but nobody paid attention, and he had to tell at homes, put on the Twitter that the data is there. So you know that even the Twitter is needed to accelerate process. And by March, there was already, Moderna was injecting the first volunteers. So, and you already know the story that end of the 2020, we already could get, or at least I did get, a celebratory manner, the vaccine and people in 2021. So many of the people who did not attended mRNA conference, from 2013, every year we had one, they didn't know that this was not the first time we were using messenger RNA for infectious disease vaccine. CureVac already 2013 started for rabies. They used at the beginning protamine, that later lipid nanoparticle for delivery. And Moderna ran already several clinical trials. Okay, there were just, as I said, participants were 100 or 200 and not 200 million. So nobody paid attention, but it was already tested out. And where we are now, you can listen, Drew Weissman can give you lectures about where we are today, that more than 250 different mRNA-based clinical trial is running right now on only the SARS-CoV-2 specific vaccine, as well as the RSV, which is FDA approved. But all of these other viruses, there are clinical trial in advanced stage. Most likely the flu will be the next one. There is also flu, RSV, as well as SARS-CoV-2, one injection already in clinical trials. So all of this respiratory, this virus against, and there are also, against bacteria, like tuberculosis or Borrelia, which cause Lyme disease, and malaria, also clinical trial running at BioNTech. And Mostly you can hear in the news about cancer vaccines, some of them known tumor antigens, those are FixVax. There are also individualized cancer vaccine where only that patient could get the vaccine because neoantigen based on the mutation present in their mRNA, their genome, and the amino acid change result, and that's also ongoing for melanoma, pancreatic cancer. This is all applied after operation and to prevent return of the tumor. And there were a trial in acute disease. VEGF-A mRNA was already well before the pandemic was tested out for wound healing as well as for heart failure. And you can hear about genetic disease. I mean, Moderna is specializing now in rare diseases, and advanced clinical trial is ongoing, and more frequent disease like sickle cell anemia or cystic fibrosis. There are clinical trials ongoing. Some is already was completed, but those were at point was not as successful. And other disease like allergies, which are not human trial yet, but for dust mite and peanut allergy to induce tolerization. I have to say, you know, many other vaccination used mRNA. Large animal like the elephant in Texas Zoo, they get 150 microgram of mRNA coding for a viral antigen. Or recently was published the shrimp, you know, get intramuscular. So this was a little bit bigger shrimp, not the tiny one. Intramuscular injection for a white spot disease, so that you will see that more will come. And what I left for me now is just telling you that I try to inspire the next generation. That is Drew and me there, but you know, these are, you know, Korean, Japanese, and English, Hungarian books about me. I didn't write those. I did write this one, and now that 11 different languages, you can purchase a book about my life, which was, you know, written during the pandemic, and so I try to describe that how you can be resilient and how you can live your life happily. Even, you know, surroundings can be very disturbing, but, you know, you can live happy, just focus on what you can do and, you know, calm down. And I mentioned here this morning, yes, sometimes under the table I showed my middle finger, but never on the top. And so that's what, you know, you have to come and enjoy because, you know, you are here, you are a scientist, you are already so privileged, and so you have to be happy. And because this week is a Nobel week, so I show you here a picture, you know, what counts. I am there with my husband in Stockholm, and in the middle section, my daughter, my sister. So the family is important. I know that being a scientist is not a family-friendly job, but you know, I mentioned my daughter, the fancy dress there in the middle. She get the best dress in the whole party, but she is a 2 times Olympic champion. So you can have children, family, if you want. If you read the book, you can learn. I never wanted. And when I looked at I was a student and my fellow student had a child, I looked at that as a big problem, just like that. But when mine was that was, you know, different story. Anyway, so, and here, you know, surrounding my daughter there is all of my teachers. And the black there is with Ava, who we did the liposome experiment here in Ernő, we did the liposome experiment together. They were my guests. And of course, David Langer and Elliot Barneten, who helped me at Penn, they were my guests at the party. And so be nice to your colleagues. You never know. Maybe you get the prize or maybe they get, but you can go to Stockholm. And with that, I thank you very much for your attention. exactly. I finished in right time, yeah.